Hello, my name is Chris Pierce, and I'm a professor associated with the Rutgers Addiction Research Center. And today I'd like to spend a few minutes talking to you about a paper that my group recently published in Addiction Neuroscience, where we focus on transgenerational memory deficits in animals that are exposed to cocaine and their offspring. So I'm going to explain what all that means in just a second. But first, I'd like to note that this is the work of Matt Wimmer, who's now a faculty member at Temple University, but he was a postdoc in my lab when this work was done. And the first thing I'd like to emphasize is this was a real synergy of research background. So my labs for many years has studied animal models of addiction. And when Matt came to my lab, he had just finished a PhD thesis in neuroscience focused on animal models of learning and memory. And so we combined our those two backgrounds into an ongoing project in my lab focused on epigenetic inheritance. So epigenetic inheritance is heritable changes in the phenotypes caused by mechanisms other than changes in the associated DNA sequence. So when we think about heritability, we think about Darwinian heritability, which indeed is associated with changes in the DNA sequence. And this takes thousands, if not tens of thousands of years to happen. Whereas in contrast, epigenetic inheritance can occur in as little as one generation. So now there's a lot of examples of this to date, both in animals and in humans, indicating that parental environmental disturbances, and there's a lot of these, including famine, toxin exposure, extreme stress, etc., can produce physiological and behavioral changes in the descendants through epigenetic changes in the germline. So the germline being epigenetic changes being the mechanisms, which are different than the mechanisms we think about with Darwinian heritability, and the germline in the parents being either the sperm or the eggs. So we were interested in developing an animal model looking at cocaine. So we're an addiction neuroscience lab, and so we were interested if parental exposure to cocaine would produce changes in behavior and physiology in the offspring. And in this case, in this study, we also looked at the grand-offspring. So in this experiment, we looked at male animals. So it was just the dads, if you will. There were rats, white rats. And so we had two groups. So we had cocaine dads, if you will, and saline dads. So the cocaine dads, we allowed to self-administer cocaine for 2 hours a day for 60 days. Okay, that's a lot of cocaine exposure, and so we really don't have to train them to do this. They like cocaine. They pick it up very rapidly. You can't really tell from the scraps. You can see they're taking cocaine, but trust me, that's a lot of cocaine over 2 months. So why did we do 2 months? So I mentioned to you that we think that the mechanism where if we see changes that are transmitted from the parent to the offspring, that they go through the germline. In this case, with the dads, it would be through the sperm. And interestingly, sperm turns over. So the sperm doesn't last forever. They live in a rat a little less than 2 months. So that's why we chose 60 days, so that we wanted as much sperm as possible to be exposed to cocaine, during this cocaine self-administration period. So those are the experimental animals. The control group are saline animals. So every time that the cocaine animals received cocaine, we had a program that there would be associated animals that would receive saline for no apparent reason. So they're minding their own business and they get an infusion of saline every time an animal somewhere in the same room as them receives cocaine. Okay, so 60 days of either cocaine or saline. And then on the 61st day, we introduced our cocaine rats and our saline rats to naive females. They were together just long enough, which is just a couple of days, for the females to become pregnant, at which point the cocaine dads and the saline dads went away, never to be seen again. And then the impregnated females gave birth to their pups. They raised the pups. The pups were weaned, and then we asked questions. We've asked them numerous questions of the offspring. So that's the first thing we do is so we have the first generation, the children, if you will, are called F1. And if you're, in this case, again, these are all rats. If your dad took cocaine, you're cocaine sired, you're F1 generation, the children, and you're cocaine sired. And if you're the control group, you're F1 saline sired. So as again, we've asked many questions over the last 15 years or so, but in this case, Matt Wimmer was interested in examining learning and memory in this F1 generation. So he used a couple of different tasks, an object place recognition task and a novel object recognition task. We're just going to focus on the first one, the object place recognition, for the sake of time today. But the interesting thing is with this task, the object place recognition task, you need a hippocampus. So the hippocampus is a part of your brain that's very much associated with learning and memory. Here's some artwork by Greg Dunn. It's a beautiful area of the brain, obviously, very intricate. And so object place recognition, which I'm going to walk you through on the next slide. For this, you need a hippocampus. And it's a very simple task. So we have an open field, a couple of feet by a couple of feet, and we put the rat in there. These are the children, the offspring, and we just let them roam around, get used to being in there in this environment. Then we take them out, and then we put in these objects, which are squares here, but in real life they were toys, and they were absolutely identical. Two absolutely identical toys, which we stuck onto the floor so that the animal couldn't move them. Okay. So then during training, we put the animals in there for a few minutes, and they're allowed to interact with these two toys, these two objects. And they're exactly the same, so the animals spend about the same amount of time interacting with the two of them. Then we take them out, take them back to the colony. They stay there overnight. They come back the next day, and then we move one of them. So the animals recognize, if they have a functioning hippocampus, they recognize that one of the two objects has been moved, and they spend more time interacting with that object. So pretty simple task, but it's very robust, and you can test hippocampal based forms of learning and memory. Okay, so there's a lot going on in this slide. So let's look down here at the bottom. So we have during training, the animals are spending about the same amount of time interacting with these two objects, which are exactly the same. So that's shown here. Training is in the white bars, and this is 50%. This is a preference for what eventually is going to be the displaced object. But you can see during training, male, female, saline sired, cocaine sired, everybody's spending about half of the time interacting with these two, 50-50 with these two objects. So then let's look at the females. The females are here on the right. So these are females whose dads either took cocaine or took saline. And you can see the saline sired animals, this is the normal behavior. So they spend, something like 70% of their time interacting with the object that was moved. So they recognize it was moved, and that's shown here. If your dad took cocaine, exactly the same. You can see these, they look almost identical. Okay. So if you're female, everything's perfect, right? If your dad took cocaine, everything's fine. Looking over on the left, you can see if you're male, not so good. So, you're showing preference if your dad took saline, if you're a control animal. But if your dad took cocaine, you have no recognition that this object has been moved. Okay, you again the animal spent the same amount of time with these two objects, indicating that they have problems with their hippocampus, and you need your hippocampus. We all need our hippocampus to make it through life every day. So just real quickly, so what I just showed you on that slide was male but not female offspring of cocaine sired rats have impaired spatial memory. That's what we just showed. Don't have time to get into all this other mechanistic stuff, but we did show that there was impaired hippocampal plasticity in these animals, and we showed changes in epigenetics. So the mechanisms that you can get these transmission of these heritable marks in as little as one generation, those are epigenetic marks, and we did show some of them, which again, we don't have time to get into that right now, but I'd like to share with you one other thing, which is we just looked at the children. So what about the grandchildren? So that was another question that we wanted to ask in this paper. So we knew it would make it to the first generation, but what about the next generation? So things get interesting, because if you see things in the second generation, in the F2 generation, it oftentimes then you see it in 3, 4, etc., indicating that you may have a permanent heritable change. So that's what we asked next. So here on the left, we have the data we just showed. So these are the offspring. So these are the male children of animals that either received saline or cocaine. And this is the phenotype we just demonstrated, which is this deficit in learning and memory. If your father took cocaine, over here, these animals, if their grandfather took cocaine. So their grandfather took cocaine. Their father, however, was perfectly normal. Never, I mean, nothing ever happened. Well, they had learning and memory deficits. We know that from over here. but otherwise they're perfectly fine. They never saw cocaine is the point I'm trying to make. Okay. But then even under those circumstances, this learning and memory deficit makes it into the grand offspring. Okay. So in the F2 generation, suggesting transgenerational epigenetic inheritance. So that's a lot of fancy words, but what does it mean? So it means in the relatively brief exposure to cocaine among these male rats results in learning and memory deficits in the male offspring and in the grand offspring. And if these changes make it to the grand offspring, it suggests again that it may then go to the great offspring, et cetera, which I think you'll agree has potentially profound implications if these same things are demonstrated in humans with cocaine use disorder. And that's the paper. If you'd like to look at some of the details, you can look that paper up. So I'd like to just wrap up by acknowledging the support from the National Institute on Drug Abuse for all of this work, and it's very labor intensive. So here's my pictures of my lab in recent years. I'd like to thank them for all of their hard work, and I'd like to thank you for listening.